We continue our study about the immune response. In the last video, we studied about the difference between non-specific and specific immune response. We remember that during a non-specific immune response, the nature of the antigen or the intruder is not identified by the non-specific immune system. The factors of the system are the macrophage and the granulocyte neutrophils. These cells phagocyte without identifying the nature of the intruder. This response is characterized by an inflammatory reaction and by a process of digestion of the intruder called phagocytosis. During the specific immune response, the identity of the intruder is well recognized by the effectors of the immune response, which are the lymphocytes. And this specific immune response can be cell-mediated, where the effectors are the T-C lymphocytes, or it will be humoral, where the effectors are the antibodies secreted by the plasma cells differentiated from B lymphocytes. So now in this video we will study about document 3 and 4 concerning the induction of the specific immune response with its two types, humoral and cell-mediated. So the transition between the non-specific to the specific immune response will be detailed in this study. First of all, we study the cooperation between the macrophage, a component of the non-specific immune response, and the Th lymphocyte, the helper T4 lymphocytes, which is the main cell of the specific immune response. In this experiment, if we remove the spleen, which is a secondary immune response, where the specific immune response occurs from a mouse, and we extract from this organ the cells, then we separate these cells into different culture. In the first culture, if we put only the macrophages, and the third culture we put only the lymphocytes, and the second one we put together the macrophage and the lymphocytes. Then we add to each medium the antigen SRBC, sheep red blood cells. These antigens are considered to be as non-self with respect to the mouse. Concerning the results, we see that in the first culture containing the antigen, non-self antigen with the macrophages alone, there is no production of antibodies, and this is normal because the macrophages are not the cell that secrete antibody. In the third culture, there is no production of antibody by the lymphocytes that are present in this culture, but in the second, where macrophages and lymphocytes are put together, the production of anti-SRBC antibodies occurred. So we can conclude simply that the coexistence of these two cells, the macrophage component of non-specific immune response and lymphocyte component of specific immune response, this contact, this coexistence, this cooperation is required in order to obtain antibodies, in order to have activation of B lymphocytes that differentiate later to plasma cell secreting of antibodies. So we demonstrate in this document that the non-specific immune response manifested by macrophages can induce or provoke the specific immune response characterized by lymphocytes. Now we will study in detail this induction or this cooperation. Here in this document, we have at the level of an infected tissue, a bacteria that enter the body. During the first contact, this is a macrophage component of non-specific immune response. This macrophage uh, start to phagocyte the bacteria, digests it, and transforms it into peptides. These antigens are, after phagocytosis, presented at the membrane of the macrophage. And they are presented on a specific site, as you know, which are called HLA of class 2, because this is an immune cell and not any nucleated cell in the body. So now the macrophage, after phagocyting the bacteria, is considered now to be an APC, antigen presenting cell. This APC will induce the specific immune response in order later to determine how this bacteria will be killed. So this macrophage migrate from the infected tissue to the secondary lymphoid organ, and here it migrates to a lymph node, to the neighboring lymph node. The lymph node is a secondary lymphoid organ. This macrophage migrate via the lymph in order to reach the lymph node. In pink, this is the lymph node. The macrophage presenting at its membrane the HLA class 2 with in red the antigen of non-self, the bacterial products. It entered the lymph nodes, and in the lymph node the lymphocytes are present. The lymphocytes Th are present. The lymphocytes Th are different because each one carries at its membrane a receptor named TCR different from the other. Each TCR is characterized by a double recognition. As seen later, each TCR recognizes two things. The TCR recognizes the non-self antigen in red. Not only, but it should recognize also the site which is the HLA class 2. So the complex non-self antigen plus HLA2 expressed at the membrane of an APC is identified by the TCR of a TH. This TH is exclusively selected because its TCR is specific to this complex HLA peptide. The other lymphocytes don't have a specific TCR, so they leave the lymph node and recirculate in the body in order to search other antigens which are specific to their TCR. This lymphocyte in blue is exclusively selected, so it is activated. So this is the activation or the clonal selection of Th lymphocytes. The first sign or the first step of the activation of this Th is the proliferation. Here we see that the Th proliferate, divide, make mitosis in order to have great number of cells, all have the same TCR, the same clone. concerning the steps of the activation of T helper lymphocytes. So when a TH is exclusively selected due to the specific recognition with an APC carrying on its membrane HLA class II plus non-self antigen. So the first step of clonal selection or the first step of activation is named multiplication or proliferation or mitosis. All these TH cells possess at their surface the same TCR. They are called all clone of TH. Among these TH, some cells transforms into memory cells that can live for a long period, for many years. And these cells can be used later if the body encounter the same antigen. And the other part of these TH differentiate, which means that they acquire a new characteristics into effector cells. The TH cell, which is the helper cell, is not a cell that kills, but the cell play a role in helping the other components of the immune system, and in precision this cell determine the ongoing of the specific immune response. If the non-self antigen, it determines, in other words, if the non-self antigen will be killed by humoral mechanism or by cellular mechanism. So when TH differentiate, they become interleukin secreting cells. So these TH after differentiation become cells that secrete chemical mediator in the blood called interleukins. Interleukins are among the family of cytokines. They are chemical substances that are secreted by TH activated and differentiated, and these interleukins, once secreted in the blood, they activate the other components of the immune system in order to determine the type of the immune response, if it is humoral or cell mediated. So in this document we will study in detail how these TH how do these TH activate or determine the ongoing of the specific immune response through the secreted interleukin. In this experiment, we have two media. In the first medium, we have an antigen called CGB, Calmette and Guerin bacillus, which is the bacteria of tuberculosis, a pulmonary disease. This bacteria is incubated or cultured in presence of macrophages and TH. On the other hand, in the second culture, we have tetanus toxoid, another antigen different from the first antigen. This antigen is secreted by another bacteria called Clostridium tetani. And this antigen of non-self is also cultured with macrophage and TH cells. So in both media, the macrophages will start the immune response. So here the macrophage would phagocyte the CGB, digest it, and present a sample from this bacteria on HLA class II and activate exclusively the TH cell. In the second medium, the macrophages would phagocyte the tetanus toxoid antigen. After digestion, the macrophage present at HLA class II samples of this antigen in order to activate TH cell. Now Th cell here and here in the two media are activated and differentiated. They would be secrete interleukins. Here the secreted interleukin is of type 2, interleukin 2, but here it's of type interleukin 4. So we can see clearly that the type of interleukin depends on the nature of the intruder in the body. In order to know what is the difference between IL-2 and IL-4 at the functional level, we transfer the supernatant from these two media. The supernatant means only the liquid fraction that contains the cytokine without the cellular fraction. So if we transfer this supernatant to another flask here and here, so here we have only interleukin 2 and here only interleukin 4 without macrophages and Th. And we add in the two media B lymphocytes and TC lymphocytes. We observe that only in the first medium the TC cell proliferate, and the proliferation of is the first step of activation. But in the second medium only B cell are activated. Since TC are the effectors of the cell-mediated specific immune response, so interleukin 2 is specific to determine how to choose that the immune response is cell-mediated. And since B cells are the effectors of the humoral immune response, so interleukin 4 is responsible for the determination of the humoral specific immune response. So we summarize according to the type of the intruder, the Th that recognize the intruder expressed at the surface of macrophages secrete different types of interleukins, and according to the secreted type of interleukin, the specific immune response can be humoral or cell mediated. Now we want to study in detail the induction of the humoral specific immune response and the induction of the cell-mediated specific immune response. Concerning the humoral specific immune response, here we have the Th cell or T4 characterized by a receptor called CD4 and a TCR which is specific to the complex HLA class 2 peptide. In yellow we have HLA class 2 and in red we have the antigenic peptide of non-self, which are, this complex is expressed at the surface of an APC or a phagocyte after phagocytosis. This Th is now activated, differentiated, so it will be secrete a type of interleukin, okay, IL. On the other hand, we have B cells. These B cells that are present at the level of the secondary lymphoid organs, lymph nodes or spleen, are of different specificity. Each one carries at its surface a certain type of receptor named BCR or antibody, simply antibody, and each B carries an antibody different from the other. Each one is characterized by the specificity of an antigen. We remember that B cells, when they recognize their antigen, we should recognize the antigen at the native state, at the free state, not expressed by HLA system at the membrane of any cell. So in this case, only this B lymphocyte whose antibody is specific to the native antigen only is selected. This is the clonal selection. This clone is selected. The other are not selected. They circulate outside the secondary lymphoid organ in order to search other antigens that can be specific for them. So this B lymphocyte that recognize the native antigen necessitates a signal coming from CD4, from the helper T cell, before undergoing any multiplication. This signal is the interleukin 4. So now, recognizing the native antigen and having a signal from Th cell, the specific B lymphocyte undergo multiplication, proliferation, mitosis to have great number of cells, clone of B cells having all the same BCR that recognize the same free antigen. Among these cells, some cells develops into memory cells that play a role during a possible second contact with the same antigen after several years. And the other B cells undergo differentiation by acquiring new characteristics in order to be effector cells. These cells, having short time life, they differentiate into plasma cell. These plasma cells are antibody secreting cells, and hence the humoral specific immune response is developed. So in order to have a humoral specific immune response, two conditions are necessary. The first condition is that having a B lymphocyte whose BCR specific to an antigen at free state, and the other condition is a signal called interleukin 4 coming from T helper cell that had recognized the same antigen presented at the surface of an APC. Now the same idea, but concerning the cell-mediated specific immune response. Here we have the same figure. The TH having the TCR specific for HLA class 2 and the antigen, non-self antigen expressed at the surface of an APC. And so this cell, after recognition, it will differentiate into interleukin secreting cells. So this cell will secrete a certain type of interleukin. Here we have in the secondary lymphoid organ a great number of TC cells, T8, T cytotoxic. These TC cells, they are not effector. They become effector under two conditions. First, we have these all TC cells. Each one has a TCR which is different from the other. Each TCR recognizes two things. It's characterized by double recognition. The TCR of TC cells, as seen in the previous chapter, recognizes specifically an antigen, here in red, non-self antigen expressed by a nucleated cell or an infected cell, and the site of expression is the HLA of class 1. Why 1? Simply because this is a nucleated cell and not an immune cell. So when a cell is infected in the body, it expresses at HLA class 1 a sample from the virus or the bacteria, and the complex HLA class 1 with the non-self antigen is recognized specifically by the TCR of a TC. This TC is specific to this complex. So this TC is exclusively selected. This is the clonal selection or the activation. The other TC, they are not implicated in this recognition, will circulate outside the secondary lymphoid organ in order to search another antigens of certain specificity. So once activated, this TC requires a signal from T4, from the T helper cell. This signal is now the interleukin 2 and not 4. So under these two conditions, the double recognition and the interleukin 2, the TC involved undergo multiplication. It gives a great number of TC, same clone having the same TCR specific for the complex HLA, one peptide, the same HLA. Some cells develop into memory cells will be used later if the same antigen enter the body after a certain time, and the other cells are now effectors. They differentiate into effector cells. The TC become effector, so it acquires a new characteristics, which is the cytotoxicity, or the capacity to kill. So to finish in this concept map, upon the entry of an antigen to the body, a virus or a bacteria, the first line of defense after the natural barrier is the non-specific immune response. The macrophage phagocyte the antigen and presented it at the HLA class 2, and the cell become now an APC, and it activates the TH lymphocytes. Once activated, these TH undergo a clonal selection. A part transform into memory, and another part into effector cells. Effector, which means they are helper by secreting interleukin. If the interleukin secreted is IL-4, so B cells are implicated in this case, and B cell that recognizes the free antigen receiving now interleukin 4 from T4, they are activated, proliferate, a part transform into memory, and another part differentiate into plasma cell secreting antibodies, and hence the humoral specific immune response is established. If the interleukin secreted is IL-2, so the implicated cells are TC. TC do not recognize the antigen in its free nature, but they should recognize the antigen expressed on HLA class 1 of an infected cell. So upon this recognition and in presence of IL-2, TC undergo clonal selection. They proliferate, a part transform into memory, and another differentiate into cytotoxic T cell or killer cell that that kill the infected cell. So in these two documents, we studied how the specific immune response in its two types, humoral and cellular, is induced. In the next documents, we will study the mechanism of antibody secretion by the plasma cell, and the mechanism of cytotoxicity accomplished by TC lymphocytes.